This is the model showing the development of neural tube and its derivatives till spinal nerve. In the first number, this yellow colored structure is the neural plate. Neural tube, before forming, it is neuroectoderm, which is surface ectoderm thickening overlying the notochord, and this thickening of surface ectoderm specialized as a neuroectoderm and called as a neural plate. This neural plate having junction with the surface ectoderm. This is neuroectoderm, this is the surface ectoderm, and which continues as a amnion. This is the notochord. This is paraaxial mesoderm. This neural plate thicken overlying the notochord now gets in number 2 grooved itself. This neural groove is deepened and junction still remains as a neuroectoderm and the surface ectoderm. This surface ectoderm continues as a amnion. This is the notochord. This is the paraaxial mesoderm, which further develops into somoid. Now, neural groove at the lateral margin of neural groove approach with each other and fuses with each other and get cut off from the surface ectoderm. So, neural tube is formed. Its formation is at the level of 4th to 6th somoid level, and then it grows cranially and caudally. Cranial opening of the neural tube is called anterior neuropore. Posterior opening of the neural tube is called posterior neuropore. Anterior neuropore closes at 20 somoid stage and posterior neuropore closes at 25 somoid stage. This is the neural tube formed. The cavity inside of the neural tube forms the central canal. This is the somoids which are formed from the paraaxial mesoderm. This is the notochord. This is the surface ectoderm. In the fourth number, we can see there is a differentiation of neural tube into ventral basal lamina and dorsal alar lamina separated by sulcus terminalis. Cavity inside is converted into central canal. This somoid is divided into ventromedial myotome and dorsolateral dermatome. This is the surface ectoderm. This is notochord. Fifth number, we can see previously neural tube is lined by germinal cells. Now it is differentiated into 3 zones. Cavity lined by ependymal layer, then there is a mantle layer, and outermost is the marginal layer. We can see also another yellow structure. This is dorsal root ganglion, which is derived from neural crest cells with its dorsal horn. So dorsal root. This is the dorsal root ganglion and ventral root and the dorsal root join to form the single spinal nerve. White colored is the single spinal nerve formed from the ventral root from ventral horn and dorsal root from the dorsal horn. Now the spinal nerve giving branches to the somite, which one is differentiated into dermatome and the myotome. This is the surface ectoderm. The fourth last one we can see the central canal. It is narrowed. The ventral horn large as compared to the dorsal or the posterior horn. This is dorsal root ganglion forming the dorsal root. This is the ventral root joining to form the single spinal nerve. It is giving the muscular branches to the myotome. It also gives the preganglionic rami, which goes to the sympathetic ganglion forming the sympathetic chain. It also gives postganglionic rami. This is the postganglionic, this is preganglionic rami. These are the muscular branches. Also the ganglion gives further visceral rami. This is again surface ectoderm.